During interphase, chromosomes are replicated in an uncondensed form, which makes them unrecognizable, in preparation for meiosis. The centrosome is duplicated, resulting in two centrosomes that are adjacent. Prophase 1 is the longest stage of meiosis, where the chromosomes progressively condense throughout the stage. After interphase, duplicated sister chromatids are held together by proteins called cohesins. First, the chromosome pairs along with its homologue line up according to the specific locus and crosses over, which is when DNA molecules of non-sister chromatids are broken apart by proteins and rejoined together. In this process, the DNA of non-sister chromatids from either parent are broken by the protein's corresponding points. Next, a zipper-like structure called the synaptomal complex holds one homologue tightly to the other in an event called synapsis, during which the DNA breaks are closed up so that each broken end is joined to the corresponding part of a sister chromatid. Crossing over results in one or more X-shaped regions called the chiasmata, and the two homologues form a tetrad. The centrosomes move towards either end of the cell where the spindle microtubules form, while a nuclear envelope breaks down. Later, the microtubules from the centrosomes, now located at the poles of the cell, attach to the kinetochores, one at each centrosome along each of the homologues. The tetrads will then move towards the metaphase plate and begin metaphase 1. Pairs of homologous chromosomes are now aligned as tetrads randomly with equal probability of either one ending on either side of the metaphase plate. This correlates to the law of independent assortment, as alleles for genes from either parent could end up in either daughter cell. The two centrosomes are now at opposite ends of the cell with tetrads aligned at the metaphase plate, which is an imaginary plate that is equidistant from the spindle's two poles. The kinetochores at the centrosome of both sister chromatids from one homologue attach to kinetochore microtubules from one pole to the other chromatid attaching to the other pole. The tetrads are now ready to separate. As proteins responsible for sister chromatid cohesion between each homologous pair break down, the homologues for each pair are pulled apart by spindle apparatus to opposite poles of the cell from the metaphase plate. However, sister chromatids for each homologue are still connected by a centromere, causing chromatids, homologues, to move as a whole toward the specified pole. Additionally, this phase exemplifies the law of segregation, which states that the alleles for a heritable character segregate during gamete formation and end up in different gametes as anaphase 1 splits up the homologous pairs, separating the possible alleles for a specific locus. In telophase, two sets of chromosomes are now separated by the formation of a cleavage furrow in animal cells or a cell plate in plant cells. Each chromosome is still comprised of two sister chromatids and regions of other homologue pair from crossing over. Cytokinesis, the splitting of the cytoplasm, occurs simultaneously and results in two haploid daughter cells. DNA is not replicated again in this stage. Some species may have their chromosomes decondense and form a nuclear envelope at the end of this stage. The spindle apparatus, which is the spindle fiber made from microtubules, starts forming and moving toward either poles of the cell. During prophase 2, the nuclear membrane in the cell breaks apart, and the chromatids that were originally scattered across the cell move towards the center, which eventually forms a metaphase plate. The chromatids that were crossed over during prophase 1 are known as non-genetically identical chromatids because the maternal and paternal chromosomes often trade genes, making the gamete diverse. Metaphase plate starts to form as the chromatids line up. Kinetochores are attached to the microtubules of opposite ends. Centromeres separate in anaphase 2 because the microtubules attached to them start pulling them to opposite poles. Sister chromatids get pulled to opposite poles, becoming their own separate chromosomes. Chromosomes decondense into loose chromatin. A nucleus is reformed in each of the cells. A cleavage furrow is formed between the two splitting cells and eventually breaks off into two separate cells. In plant cells, a cell wall is formed. In the end of meiosis, there are 4 haploid daughter cells formed, each genetically distinct due to crossing over. After meiosis, 4 sperm cells are formed. For women, 3 of the 4 eggs get destroyed, and in the end, only 1 egg is released into the fallopian tubes.